Welcome back. Today we are going to learn hybridoma cell culture, and in hybridoma cell culture a particular topic we are going to learn is like production of monoclonal antibodies and their applications. This topic is there in B Pharmacy finally year in pharmaceutical biotechnology subject. So here we are going to learn about the definition, history, discovery, and the method of preparation and their applications. Hybridoma technology is a method where we produce large number of antibodies. These antibodies are called as the monoclonal antibodies. Now, what is the term hybrid? Now, a diploid organism or a cell which is a result from a cross between two dissimilar parents or the cells is called as a hybrid. Now we are learning about the monoclonal antibody production, right? So now what is this monoclonal antibodies means? The antibodies which are produced by a single antigen forming cell or a clone which is directed against a single antigen or antigenic is determined to be a monoclonal antibody, and the example is the plasma cell tumor, that is the myeloma cell. Okay, now what is the history of this hybridoma technology, or the as we taken the example of myeloma, we have a myeloma theory. So in 1970s a B cell cancer myeloma was discovered, and it was understood that these cancerous B cells are produced by a single type of antibody, and this is where the study of the structure of the antibodies has begun. Now, what is the discovery? Now, the magic bullet, the hybridoma technology, was first proposed by a scientist known as the Paul Ehrlich in the beginning of the 20th century. Okay, and then it was followed by George Kohler and Cesar Milstein in 1975. They have shared a Nobel Prize in Physiology and Medicine in 1984 for discovery of hybridoma technology with Niels Carr's journey. Okay, now these hybridoma, the term was coined by Leonard Herzenberg in 1976 to 1977. So now, what is the principle of this hybridoma cell technology? Now, we said it is an antibody producing cell or an antibody, right? So now this antibody producing a B lymphocyte is collected from the spleen of a mouse, which is immunized with the RBC from the sheep, and a single myeloma in a bone marrow tumor cell which is capable of multiplying are fused to produce the hybridoma cell or a hybridoma. That means the antibody lymphocyte from the spleen of the mouse is immunized with the RBC from the sheep, and a single myeloma in the bone marrow tumor cell which is capable of multiplying, and then they are fused together to produce a hybridoma cell. Now, what are these hybridomas? What is the use, like what they are special enough to be studied of? Like these hybridomas have antibody producing capability. AB is nothing but antibody producing capability, and they have the ability to grow continuously like the cancerous cells, and they are also immortal like malignant cancer cells. See, the myeloma cell divides and the B cell produces the antibodies. Okay, now the method of preparation of hybridoma technology has overall 8 steps. The first step is immunization of mouse, isolation of B cell from spleen, cultivation of myeloma cells, fusion of myeloma and B cells, separation of cell lines, screening of suitable cell lines, followed by in vitro or in vivo multiplication, harvesting, then transferring it to production of drugs, then introducing the drug or the particular product into the market, that is a pharmacovigilance, then it is carried on. So we'll just study these 8 steps clearly so that you can understand how the hybridoma cells are produced. Now, immunization of the rat. So what a mouse is taken which is immunized with the pure selective antigen for the production of specific antibody due to proliferation of desired B cell. Okay, a specific rat is taken and then it is isolated and immunized to produce a specific antibody. Now, after immunization of this rat, we isolation of the B cells. Second step is isolation of B cells. Now, when this rat produces a good response, good immune response, then this rat is killed and the B lymphocytes are harvested from the spleen or lymph node of the rat, and this harvested spleen or lymph node is made into a suspension. Okay, and this is kept aside. Now we have the B cells, and then we need the myeloma cells. So as I said, we need the myeloma cells and B cells. Myeloma cells helps in dividing, revision, and B cells helps in producing the antibody. So now we have the B cells from the rat. Now we need to produce the myeloma cells. Okay, now cultivation of these myeloma cells. From the mouse, the myeloma cells are collected and then they are grown in a tissue culture medium which is deficit in HGPRT. HGPRT is nothing but hypoxanthine guanine phosphoribosyl transferase. Now this HGPRT is a gene which is required for the synthesis of enzymes. Okay, and myeloma cells, these myeloma cells doesn't have the HGPRT gene and hence they doesn't produce the antibodies. Okay, now myeloma cells without HGPRT, they doesn't have the capability to produce the antibodies, whereas the spleen B cells which are collected from the mouse, which from the immunized mouse, has the HGPRT and they have the capability to produce the antibodies. Okay, now the fusion of these myeloma cells and the suspension of the spleen or lymphocyte B cells. Okay, now these both are fused in the presence of the PEG. Okay, what is PEG? PEG is nothing but polyethylene glycol. Okay, now first we have immunized the rat, isolation of these B cells from the rat, they are kept aside. Then after that, cultivation of the myeloma cells, then they are kept aside, and these both are fused together in the presence of PEG, that is a polyethylene glycol. Now after fusion, they needs to get separated. Okay, now these myeloma cells which are fused with the spleen, which are fused with the B cells, they form the hybrid cells. Which are not fused, they die. Okay, now the myeloma cells which are not fused with B cells, they die and they are removed, and the fused cells are called as the hybrid cells. Now these hybrid cells are grown in a selective culture medium that contains the HAT. HAT is nothing but hypoxanthine amino protein and thymidine. Okay, and after fusion they are separated and they are grown in n number of cells for 10 to 14 days. Okay, the fused hybrid cells are separated into n number of wells. So as you can see, like these are called as wells. Okay, the suspension is the hybrid cells are poured into that wells and they are grown in those wells for 10 to 14 days in the presence of HAT, that is hypoxanthine amino protein and thymidine. Now such tissue cultures are called hybridomes or immortal clones of cells, and they are capable of producing the MABs, that is the monoclonal antibodies. Now HAT is nothing but a medium which contains the amino protein. Okay, the name itself resembles amino protein, which blocks the nucleotide synthesis pathway. Okay, this HAT medium will block the nucleotide synthesis pathway. Now hence the cells are dependent on the another pathway, that which needs the HGPRT. Okay, and HGPRT is not present in the myeloma cells. Now myeloma cells that are combined with myeloma cells and B cells, no fusion, will die, and the myeloma cells with B cells which are fused will form the hybridoma. Now these hybridomas need to be screened. Okay, now after the growth, the supernatant of each of these liquids are taken out and they are tested for its antigenic activity by the test called as RIA or ELISA. Now the clones or the supernatant from each of these wells which are tested by these tests, which produce a desired reaction or a standard reaction, those are selected as the antibodies. Okay, now after selection, the screening is done, then we need to multiply it and then harvesting. So the multiplication is done either in lab or in the animal, that is in vitro or in vivo. Now the good antibody which are produced is found, and the MABs, that is the monoclonal antibodies, are produced either in vitro or in vivo. In vivo, it is done by injecting them into the peritoneal cavities of the mouse, and now these grow as a tumors, and they produce large quantities of antibodies in the asiatic fluid of the mouse, which can be aspirated from mice many times over the lifetime of the animal or the living being or the animal until it's died, until it's until it get died. Okay, sorry, until it dies. And now as these cells are derived from a single type of hybrid, they can produce only one type of antibody, hence the name is called as monoclonal antibodies. Mono is one, so they are derived from a single type of cell which produces only single antibody, hence they are called as the monoclonal antibodies. Now, what are the characteristics of these antibodies? Why these are made so specific? Why they have drawn the attention towards them is like they can be frozen for prolonged storage, and these hybridoma also carry two mutational defects. One is, as these hybrid cells no longer produce their own immunoglobulin as that of the normal B cell parent, and as these hybrid cells, they lack the HGPRT enzyme, which is necessary for the nucleic acid synthesis, is provided by the B cell component of the hybridoma. What are the properties of this hybridoma, like these hybridomas which are formed here, right? So they are immortal, like they can be grown limitlessly. The capacity of their growth is limitless, and they are immortal, and they can synthesize unlimited amount of IGs, that is antigens, that is like approximately 10,000 molecules per cell per minute. That means per minute one single cell produces approximately 10,000 molecules of the IGs. And there are interspecies hybridomas are also being developed, like rat and mouse, human and mouse, and recently human to human hybridomas have also been generated. Okay, now we have the serological applications of these monoclonal antibodies or the MABs. In serology, we used it in ABO blood groups, identification of the ABO blood groups. Then in diagnosis, we use for detection of pregnancy, detection of pathogens, and similar molecules can be separated by using these MABs, that is monoclonal antibodies. And the other application in serology is immunopurification. Okay, the purification of individual interferons by using MABs, and also inactivation of T lymphocytes which are responsible for rejection of organ transplants. And not only that, they have serological applications, they also have the applications in therapeutically. So the therapeutic applications are like removal of tumor cells from the bone marrow, and treatment of patient with malignant leukemia, lymphoma, carcinoma, sarcoma, and miscellaneous antigens like HLA, which are associated with A, B, C, and DR, etc. And in future, the hybridomas can be stored in frozen state and revived subsequently. And they are also marine antibodies, but these are unsuitable for human therapeutic use because they have the strong immune response induced in the man. And we also have the human monoclonal antibodies now being prepared by fusing the particular antibody fragment genes in bacteriophage genes. Okay, and the most widely used MAB is OCT in the treatment of acute renal allograft rejections. If you are preparing for some competitive exam, then you can expect this in a multiple choice question. As any product which is released into the market because of this therapeutic effects, but they also have some disadvantages, right? So some of the disadvantages of these MABs or the monoclonal antibodies is like, as it is a very rigorous process, they need more time and effort and also commitment for very large time. And the average affinity of the MABs is less than the polyclonal because these are monoclonal, and when compared to the polyclonal, the action is comparatively less. And monoclonal antibodies against the conformational epitopes on native proteins may lose reactivity with antigens, and some antibodies sometimes display unexpected cross reactions like unrelated antigens and immune rejections. Okay, so these are few of the disadvantages of the monoclonal antibodies, and the entire topic the reference book what I have used is like the textbook of microbiology from Pre Chakraborty, and the other is pharmaceutical microbiology by N.K. Jain, and some of the topic is also covered from the Wikipedia of Hybridoma technology. And the conclusion of the topic is like the monoclonal antibodies are developed with a primary objective to develop the best in class prescription products for the therapeutic antibody market, which is the fastest growing segment of global drug sales. And thank you. So he's Paul Ehrlich, who is the discoverer or the one who proposed the technology of the monoclonal antibodies. So I hope you got this. So just let me give a small summary. Isolation of the B cells from the immunized rat, and also cultivation of myeloma cells in the HGPRT medium, then fusing these B cells and myeloma cells in the presence of the PEG, then separation into the cell lines, and then they are left for 10 to 14 days. After the growth, they are screened by ELISA or RIA test, then they are grown in the in vitro or in vivo environment, and then they are made as a drugs and marketed.